Get ready to blast off into the incredible world of brain-computer interfaces. Neurocareers doing the impossible is taking you on a journey to meet the fearless pioneers pushing the boundaries of what's possible. In this special series, we'll be shining a spotlight on the nominees and winners of the International BCI Award, one of the biggest and most prestigious awards in the BCI world. You'll hear from BCI professionals as they share their revolutionary work and get a behind-the-scenes sneak peek at what it takes to be a winner. So, buckle up, grab a snack, and get ready to be amazed as we explore the impossible becoming a reality in the world of BCIs. But before we blast off, I want to give a big thanks to the co-host of this BCI Award edition on our podcast, Dr. Christoph Guger and G-Tech Medical Engineering. When I was a faculty member at the University of Cincinnati and Cincinnati Children's Hospital Medical Center, my team and I did some pretty amazing stuff for patients needing epilepsy surgery. We would put special sensors, also called grids, directly on their brain to prepare for surgery and create maps of their brain in a much faster and safer way than is usually done. It's called high gamma mapping, and it lets us figure out in just a few minutes the essential parts of the brain that need to be spared during surgery. Can you imagine? You can create a map of language, motor, and even math processing in no time. It was the first time this innovative technology was used at Cincinnati Children's Hospital Medical Center. When I moved to Florida, I established the first functional brain mapping and BCI program at Florida Hospital. I continue to use high gamma mapping to help epilepsy patients avoid losing their ability to speak or move after surgery. But even cooler than that, I created brain-computer interface studies that let patients control things with their brain in real time. Our patients could even spell words with incredible speed by just using their brains. No hands involved. I also discovered that brain-computer interfaces could help patients move their chronically impaired hands or legs years after a stroke when not much else could help them. So, I started working with the faculty from Advent Health University to help these patients restore their ability to use their hands. I received special training for it, and it's still mind-blowing how cool it is. My favorite part of what I do is teaching. At my established Institute of Neuroapproaches, I've integrated all my experience and knowledge into a unique course on brain-computer interfaces. With graduate neurobiology students, we started with theory, and then I gave them the BCI equipment so they could have hands-on experience working with BCIs. It significantly improved the way students learn, and they absolutely love this practical part of learning how to use neurotechnology. And all of this was made possible thanks to G-Tech's awesome brain-computer interface technology. They have everything from high-tech up to 1024-channel EEG systems to wearables, tools for neurorehabilitation, such as the medical grade system recoveries and educational kits with unicorn hybrid plug for learning how to work with BCI technology. But the most important part is their support and care. I have enjoyed working and collaborating with Dr. Christoph Guger and GTech employees for over 15 years, and I hope for many more successful years ahead. So, if you are interested in GTech's brain computer interfaces and neurotechnologies, check out their website at gtech.net. It's worth it. Hello, Nick. Hello, Maitreyee. It's wonderful to have you today on our podcast. Thank you very much for coming. And can you please introduce yourself to our listeners?
Thank you for having us. I'm Maitreyee. I'm a postdoctoral researcher at UC Davis in Neuroprosthetics lab, and I'm currently working on speech neuroprosthesis. And my focus is to synthesize voice from intracortical neural data.
And I'm Nick Card. I'm also a postdoc in the same neuroprosthetics lab at UC Davis, which is a part of the BrainGate consortium. And my research is also focused on speech neuroprostheses, but more of a brain to text pipeline rather than brain to voice like Maitreyee.
And we will see both of those lines in your study, which I think is fascinating. So you will be complementing each other. How did you get to do this absolutely amazing work? Maybe you can bring us in time into the past and tell our listeners how did your interest in neuroscience, neurotech develop? And how did you get to this stage that you can conduct such amazing studies?
Well, you're the senior postdoc.
Right. So my journey started in the UK. So I did my undergrads and PhD in the UK. I got interested in brain-computer interfaces when I was in high school. So I really wanted to explore that further, and I found a university with great research program, which I enrolled to for my undergrad. So I studied at University of Reading, which gave me plenty of opportunities to get involved in BCI projects early on in my undergrad. So I did summer projects on non-invasive EEG-based BCIs. Later on, I continued with my PhD studying motor systems with non-invasive BCIs. So I worked on BCIs for stroke rehabilitation, looking at neural mechanisms of movement generation through EEG. And that is how I started my BCI journey. Later on, I did my postdoctoral research at Imperial College London, again in the UK, where I diversified a little bit and I worked on conversational agents to help people with dementia and looking at affective robotics. So this combination of working on speech-related technologies and BCI technologies really fascinated me. And this was a perfect opportunity that was presented to us by Sergey and David, who were starting their new lab at UC Davis as part of BrainGate Consortium. So their focus was studying speech neuroprosthesis and making BCIs for generating speech, which was really fascinating to me. So I presented my interest to them, and then, I joined the lab.
Thank you very much. And Nick?
Yeah, so I have sort of a similar story, but from within the United States. Instead, I went to undergrad at the University of Pittsburgh, and in my freshman year, for the first time, I heard about brain-computer interfaces, and I thought that sounded pretty cool. And I managed to start as an undergraduate researcher with Aaron Batista there, studying neuropopulations and how those neuropopulations encode movement, and doing this with primates and using brain-computer interfaces. So I spent 3 years as an undergraduate researcher in his lab, and then I stayed at Pitt for graduate school, and I still studied the motor system in primates, but it was a little bit a step away from BCIs, more of a step toward basic science and tracing circuits and that type of thing. And I knew that that experience would be very valuable for me for learning about how these systems work, but I also knew that after grad school, I wanted to try to go back to the BCI field and leverage that knowledge and make an impact. So I heard that Sergey Stavisky was starting a new lab out here as part of BrainGate, and I sent him an email, and he interviewed me, and it worked out from there. And between Sergey and David, who are the two co-principal investigators of this lab, I mean, you couldn't ask for a duo with more potential. It would be hard to find, I think. And that's certainly proven true since being here. So I joined the lab just a few months after Maitreyee. And that was like a year and a half ago, something like that. Yeah.
And this was a unique opportunity to join a brand new lab. So we got to really help and build it up, build everything, all our systems, all our devices from scratch. So it was a very unique experience and very rewarding as well.
Of course, to start everything from the beginning, I think there is no better opportunity. And what do you think made you both suitable candidates for this position? I'm sure there were other applicants, but from those applicants, you were selected. What do you think were the skills that helped you to get here?
I think for me, Sergey has mentioned this, and he would know better than I do, but there's, I guess, somewhat of a trend of people going from monkey research to human BCI research. And in his opinion, those people were successful. So I think that probably helped me in their selection of me. But at the end of the day, I think it's all just hard work and putting a lot of time in and, trying to demonstrate in your undergraduate and graduate research that you're competent and hardworking, putting out good publications, thinking critically, that type of stuff. And then there's also a lot of luck involved too. When I was looking for a postdoc, this lab was starting and trying to hire a postdoc. And there's a lot of really great candidates whose timing does not work out quite so luckily. And I'm very fortunate that it did for me.
Yes, yes. It's very important to be at the right place at the right time and have right skills. Yeah, so you had all together.
Yeah, I was lucky to benefit from a lot of great training during undergraduate and graduate from great advisors. And I think that really can't be understated how important that is. To become a good researcher, you have to be trained by good researchers who are also kind and patient and that type of thing.
Yeah, absolutely. Absolutely. Thank you, Nick. What about you, Maitreyee?
Yeah, I think as Nick said, it is being at right place at the right times because as one of the first member of the lab to be hired, and I think what they were looking for was someone with more kind of diverse experience in transitional research. And I had worked with different types of BCIs before, mostly non-invasive and also speech technologies. So I had kind of right combination of these different backgrounds. I also worked with several patients with different neurological conditions, so with stroke, dementia, and Parkinson's. So probably my research experience in that area also helped. I think it was also that I applied my research on non-invasive BCIs to some of the data that Sergey has generated while he was a student, and then showed that, yeah, I can demonstrated my transferable skills. So that might have also contributed in my hiring here.
Mm-hmm. So was this data available, freely available, and you analyzed it? Is that approach that you used?
Yes, I was really fascinated to explore single neuron resolution data. When I was working with EEG, I wanted to see whether the methods that I developed can also work on the neural data that we record from a very different scale. And that's why I started exploring that data. And it turns out, yes, the methods that we develop are quite transferable. We can investigate different types of neural recordings using those methods. So that was interesting. And yes, open data sets have really helped me in my research throughout my undergrads and PhD and even today. So data sharing, looking at open data sets is really important to prototype different things and try different methods.
Yeah, absolutely. And then add this code and the solution that you used into your portfolio. Yes. So absolutely, absolutely. Now you mentioned this Brain Gate Consortium, yes, and the lab that was just being established when you came. And indeed, we know some labs in California, Eddie Chang's for many, many years. Your lab sounds very new, not very familiar. So can you tell a little bit more about this BrainGate Consortium and the lab? What are the main goals of the lab? What type of work is going on, and what are the future plans for the lab?
BrainGate currently is a collection of 4 labs. So there's Brown University, there's Stanford University, and now there's UC Davis and also Emory University. And for a long time, it was just Brown. And then Stanford joined in with them. And then Davis and Emory are the most recent additions. And each lab kind of has its own thing that it's focusing on. So Brown historically has been really focused on getting really good cursor control out of brain-computer interfaces. And they also started the consortium in the clinical trial as we know it. So they're all obviously an important player. Stanford, which is the second oldest lab in there. So the Stanford lab is led by Jamie Henderson and the late now Krishna Shenoy. And I think, at least recently, really what they've been focusing on as far as BrainGate goes has been creating BCIs with ever increasing communication rates. So that went from basic cursor control to the handwriting decoding that Frank Willett published a couple years ago. And most recently, last year, their speech neuroprosthesis work, which was one of the first great demonstrations of this closed-loop intracortical speech neuroprosthesis. And now here at Davis, obviously, we're really focused on speech, laser focused, and Emory is also focused on cursor control and using some abstract machine learning techniques to do interesting things with these types of data. And they're also now dipping their toe into speech as well. It's a growing trend. I also want to note that Sergey Stavisky, one of the co-PIs of our lab, did a postdoc in the Stanford BrainGate lab, and he was also a research engineer before that at the Brown BrainGate lab. So there's sort of a scientific tree that flows through these.
And David Brandman too.
Yes,
Yeah.
of course, David Brandman, PhD, and Brown.
Yeah. So this year, BrainGate is 20 years old, the consortium.
And when published?
About 2 years ago. So Sergey and David joined UC Davis as assistant professors in end of 2021. And then we started in 2022.
And you mentioned that already not only one lab, yes, within the consortium, already 2 labs are working with the speech neuroprosthesis. Why is such interest in speech neuroprosthesis? How valuable is it? How much in demand are speech neuroprosthesis?
Well, it's sort of a relatively recent trend. Interestingly, BCIs, especially in BrainGate for a long time, have focused on arm and hand control, and whether that's controlling a cursor or a robotic arm or some version of that. And I think there was just sort of this conception in the field that decoding speech would be way harder than doing something lower dimensional like arm and hand, because you've such this high dimensional space of speech articulators. And only recently have people actually started to try it, and especially leveraging a lot of this machine learning and large language models that have been coming up in the last few years, that's been helping to make it a more attainable goal. So I think it's always sort of been on people's radars of like, this would be great, we'd love to do this. And only in the last few years has it really begun to explode in popularity. And the impact is, I would say it's obvious, but it's for people who have dysarthria or anarthria. I mean, not being able to talk to people around you, your family and loved ones, or your work, or your friends, like, it's debilitating in every sense of somebody's life to not be able to communicate. And what was previously considered to be a pipe dream of restoring that ability is now within reach, and it's becoming obvious. And already, in our lab, we've seen a really positive impact with the clinical trial participant that we've been working with.
Yeah. And how many patients across the world do we think are in need of this type of speech neuroprosthesis?
I believe that there are millions of people around the world with some form of dysarthria or severe dysarthria or anarthria, the complete inability to speak. So people who've suffered from strokes or people with neurodegenerative diseases like ALS, or people with like traumatic brain injuries, Broca's aphasia, I guess. There's a lot of conditions that collectively add up to a lot of people that could benefit from this type of technology.
Thank you so much. And you mentioned that there is, I would say, a boom, an explosion of studies related to speech neuroprosthesis, speech decoding. Can you maybe give our listeners a brief overview of what's already available in the world so that we can already compare it with your new study?
So traditionally, people in need of some assistive technology to communicate have used eye tracker controller or gyroscopic head mouse or speller boards and other kind of technologies. But those are very tedious to use and the communication rates are very slow. So they have to select either character by character or select objects or pictures on the screen. And it can be very difficult and restrictive to communicate using those technologies. So there was a drive to kind of explore whether BCIs can fill this gap and whether we can decode speech directly from neural signals, because the participants that we are targeting have intact functioning in their cortex. So we can record the neural signals from their cortex directly, and the signals are not being able to be sent to their articulator muscles, and that's why they're not able to speak. But if we can intercept those signals directly at the source in the neural data, we can maybe decipher speech from that. So that was the motivation behind exploring BCIs for communication and speech. So we have seen some early research where labs have used ECOG to decipher characters and limited vocabulary and words from neural data. And that was kind of an indicator that we can decode brain signals to identify speech markers. And then within BrainGate Consortium, we have access to very high resolution neural recordings from Utah arrays. So these are microelectrode arrays that we implant directly into the cortex. So they are able to record single neuron resolution of information. So that can give us high dimensional information about speech correlates and the neural activity. So we found that, yes, it is possible to look at neural data and decode speech reliably. And that is what was the motivation behind our lab. We wanted to do it better. We wanted to target better brain areas that encode speech features. And we wanted to develop better decoders to decode speech from brain signals.
Well said. You asked specifically about available technologies. And as Maitreyee pointed out, a lot of the available assistive communication technologies right now are very limited in their speed or their efficacy or how naturalistic they are to use. And the type of brain-computer interfaces that we're talking about are still mostly confined to these clinical trials within academia. And only, in the last year or two have companies like Neuralink or Synchron started to explore extending these types of technologies out to more people. So Neuralink has just introduced their first participant, and they're hoping obviously to extend their technology to many, many more people. And Synchron similarly has demonstrated with a handful of people as part of their clinical trial, again, with the goal of really translating beyond that. So we are sort of in the early days of this type of thing being available to anybody who needs it, but we're also further along that timeline than we ever have been. And there's been more progress in the last year toward that goal than there ever has been. So it's a bright future.
Yeah, and that as we have new devices to record better neural signals from the brain, more reliable neural signals with higher signal to noise ratio, we will be able to deliver better neuroprosthetic systems to the users in need.
Yes, absolutely. And I would like to discuss a little bit different approaches that are being used to decode speech. I know that some of the studies, they are decoding letters, let's say single letters, some phonemes, and some single words. There are studies, actually one of the also BCI Award nominees, that are trying to decipher semantics from the brain data. Yes? So can you tell our listeners the differences between those and which one of those approaches you are using in your studies?
When you start thinking about a speech BCI, it sounds really intuitive to think that you should decode words that people are trying to say. That seems like the base unit of language. The problem with that is that any given person knows 30 or 40,000 different words, and to adequately sample all of those and train a model to do that is very difficult. So you could probably train it on 1,000 words, but then generalizing beyond that, I think would get really hard. So the advantage to using smaller units of language like characters or phonemes, like you mentioned, is that there's only 26 characters or there's only 39 phonemes in the English language. And if you can teach a model to predict these smaller parts of words, then hopefully you can get enough data and train it, and then it can generalize to any word that you might want to say as long as it's built out of phonemes and characters, right? And then the semantic is also an interesting one and probably would have strength in the ideal speech neuroprosthesis. But we focus on, or at least I focus on for brain to text generation, focus on phonemes and decoding phonemes. So as I mentioned, there's like 39 phonemes, which are sounds that make up the entire English language. So any word you can think of is just a sequence of some combination of these 39 phonemes. And if you can train a decoder to predict a phoneme from neural activity, then hopefully you can say whatever word that you want. And in our experience, that's been very much true. Out of a 125,000 word vocabulary, we can predict pretty accurately what somebody's trying to say, even if it's a word that they haven't ever tried to say with the speech neuroprosthesis before. That is our current approach. And I think realistically, like I mentioned, integrating some sort of semantic information would probably be useful, but language is a hierarchy of all these things. And beyond phonemes and words and sentences and semantics, there's many other layers that would probably be advantageous to include. And it's just a matter of starting to explore those types of things.
And we are also taking a step further with speech PCIs and trying to decode voice directly from neural activity. So, as Nick mentioned, we are able to accurately decode these fundamental units of sounds, which are phonemes, and then string them together in a sequence to form words which appear as text on the screen. But natural speech is much more than that, right? So, when we speak, we have changes in our intonations, our cadence, our pitch, expressions that we put into speech. So, in order to restore naturalistic speech, we are exploring how we can decode this neural activity directly into sounds or voice, such that when a participant attempts to speak, we can decode that in real time, produce sounds, and play it back. So they can have conversations in real time as we are having right now. And we don't have to wait for the participant to say something for it to appear on the screen and then the communication to happen that way. So we are also exploring kind of next level of speech neuroprosthesis, which is much more challenging because, again, we do not have discrete classes for voice. You can speak in any way, in any manner you like, in any pace you like. So it becomes additional challenge to synthesize voice that way. But we are also exploring that avenue as well in our lab.
Thank you very much. And now let's get directly to the study that won an award, the first place in the BCI Award Competition 2023. So can you just briefly introduce the study and tell what was the main goal for you in that study?
Yeah, so as I mentioned, we're part of this BrainGate consortium, which runs a clinical trial. So the UC Davis site of BrainGate, we recruited a 45-year-old man with ALS to our clinical trial. And, he's effectively paralyzed from the neck down. It's a terrible degenerative disease. He's severely dysarthric, so it's very, very difficult to understand him, even for an expert listener. So in his day-to-day life, his communication is very restricted to either speaking to a highly trained interpreter or using a gyroscopic head mouse to move a cursor around on a computer screen and very slowly type out sentences. So with either of those modalities, he can communicate at about 6 or 7 words per minute, which is not very fast. So our goal was to create this sort of multimodal speech neuroprosthesis that could translate his neural activity into the words that he was trying to say, whether that's into text on a screen or directly into audio that sounds like his voice.
Thank you. And as I understood, each of you was responsible for one part of this project, yes? So, Nick, for you, it was conversion into text, and Maitreyee, it was translation into direct voice. Can you tell a little bit more about those two parts and why each of you chose that particular part of the project?
Yeah, so this conversion into text, we call it brain to text, and it's not necessarily a new concept. There were 2 Nature papers that came out last year demonstrating that you could do this, and they got somewhere between a 20 and 25% error rate, which means that 1 out of every 4 or 5 words that they decoded was wrong. I mean, the sentence I just said had a lot more than that. And if you imagine that 1 out of 4 words is wrong, it would have been totally incomprehensible. But, we were still able to really benefit from their experience and what the results that those studies demonstrated, especially Stanford, who's a close collaborator of ours. So our goal specifically in this brain to text was to implant more electrode recording arrays into higher yield parts of the brain. And effectively, the eventual goal is to very accurately decode what the participant was trying to say. So, through in this highly collaborative project, I don't want to say like I'm the only text and she's only voice. I mean, we're both projects are highly collaborative, but we lead our respective projects. But in this text project, we were able to, so we went in on the first day, and this is 25 days after the surgical implant into our participant, a man with ALS. And so we wait 25 days just for healing and so that the arrays could stabilize in his brain. So we came in on the very first research session and we collected about a half an hour of data of him speaking overtly, attempting to speak prompted sentences. So what that means is like a sentence would appear on the screen and he would try to speak that aloud. And while he's doing that, we're recording his neural data. And after so many of these sentences, we then trained a machine learning model to try and map his neural activity to the phonemes that he was saying in those sentences. And then using that trained machine learning model, we then, did another block where he's attempting to read prompted sentences, but this time we were predicting what he was trying to say in real time using that machine learning model. And we were showing those words on a screen in front of him. And so on this very first day, we didn't know how good things were going to be, so we limited ourselves to a vocabulary of only 50 words just to keep it simple. There was a million things that could have gone wrong, from our decoder completely breaking to just having electrical noise in the signals or, a tornado rolling through or something like that. But we were really pleased to find out that in these very first blocks where we're predicting what he's trying to say, we were able to do it almost perfectly with 99.6% accuracy or so. We were able to predict which of the 50 words he was saying in sequence. And that was a really exciting result. We were stunned. So we came back 2 days later in the second research session. And instead of just doing 50-word vocabulary, we jumped straight up to the biggest vocabulary that we had, which is 125,000 words. And again, we collected a little bit more of this training data where we show him sentences and he reads them aloud. And after an hour or two of that, we trained a new model. And then with this huge vocabulary, we were able to predict what he was trying to say with under a 10% word error rate, which is kind of amazing in contrast to some of those studies that were just coming out at that same time, which were only able to get to about 20 or 25% word error rates. So the fact that we could do this in our very first session showed us that there was huge potential in this approach. And we were really pleased to be the first people to demonstrate that this potential was there.
Yes, absolutely. Absolutely. Maitreyee, do you have anything to add to that?
Yeah, so we are really focused on looking at all the avenues of speech neuroprosthesis and how we can make those better, and really explore how much information can we get from the brain signals that can be translated into speech. So we already had some evidence, as Nick suggested, from our collaborators at Stanford from BrainGate, who had demonstrated that it is possible to decode text, and we did it better with Nick's project. So the second thing we wanted to look at is to go a step further and see whether we can also decode voice from these same signals. So our participant was again speaking the prompted sentence, and we wanted to see if we can synthesize the voice in real time as he's attempting to speak. So the additional challenge we face here is that we don't know how he's saying the sentence or what he's saying, because his speech is really dysarthric. And the way we train these BCI algorithms is using machine learning. Specifically, we use supervised machine learning, which requires you to show it an example of neural activity and an example of the speech that is corresponding to this neural activity, and then train that algorithm that way. But we did not have an example of his voice because he couldn't speak intelligibly. And so the major challenge we had to overcome to kind of synthesize his voice is to develop new techniques to generate the synthetic voice that is aligned to his brain activity, so we can estimate what he must be saying at this particular time point with very high resolution and kind of match his brain activity to what his voice might sound like. And overcoming that challenge, we also demonstrated that we could synthesize voice, which is nearly intelligible. It's not 100% there, but we have made huge progress in that area. So he is able to say what we want with the pace that he wants, and it sounds that way to us. So we can actually hear his pauses, his emphasis on different words, and this is a step towards making these BCIs more expressive, more natural, and bring them closer to more human communication. So we also tried this on the first day. We collected the data. Now, the data that we collect, we can pass it on to different decoders and try to identify different things from it. So Nick used the same data to identify text and words from the same data. I tried to synthesize voice. We tried it on the first day, from first day, and then we're able to hear for the first time the voice synthesized from the neural activity in real time. And that was also very exciting for all of us. And both of these applications are the first, we believe, are the first demonstrations of BCI in action on the very first day. So this kind of helps us to estimate that if this technology becomes available to larger populations of patients, how much time do we need to invest for these technologies to be useful to them? So initial estimate was that we have to collect days and days worth of data before they start working. But now we know that at least in the minimum capacity, they can start working immediately after hour or half hour.
Mm-hmm. Thank you so much. And what do you think made your study to provide these amazing results that were not possible in previous studies and very recent previous studies, yes, who reported still 25% error rate and so on and so forth?
I think there's a lot of reasons. So one is we have the benefit of seeing what these other studies have tried and how successful those approaches were. Whereas they really pioneered their way into this field, not knowing what they would find and developing the methods accordingly. We have that starting point to go from, which is a huge help. And I think another major factor is just luck again. That, like we mentioned luck before when looking for postdocs, and it's not only luck, it's luck that's engineered by skill, but there's a lot of things that can go wrong in a clinical trial, in a surgical implant, in a day one experiment. Even if everything goes right, you can have a participant that's just not a hard worker and doesn't really want to do it and isn't engaged. But in every facet of this, I think things have worked out really well for us. So it starts with David Brandman, who's one of the co-PIs in this lab and an expert neurosurgeon. And he meticulously, well, first of all, he identified this man with ALS and enrolled him into the clinical trial. And second of all, he meticulously planned the array implant surgery and where we were going to target the arrays and executed it with perfection and has provided wonderful boutique neurosurgery care to the participants since then. And then, on our ends, we are in the background developing our real-time data collection rig, and through a lot of hard work and collaborative efforts, we have developed a really nice, flexible platform for us to collect data and run experiments with. And then it gets to the point where everything's ready and you show up on the first day. And the first time you plug in these arrays, you don't know exactly what you're going to see because there's only so many people in the world that have been implanted with these, and there's huge sources of variance in neural recording quality that aren't completely understood right now. So you don't know how well the surgery went from that perspective, and you don't know, like there could be an Xbox in the corner of the room that's sending off a bunch of electrical noise, and it's going to mess up your recordings or something like that. So there's a million factors, and in a nutshell, I think we got really lucky. Our hard work paid off in many ways. We were able to implant twice as many electrodes as the previous Stanford study was into the premotor cortex, and we targeted some new areas there that we thought would contribute heavily to speech, but we weren't sure because they haven't been recorded from before. And they do. They are very good contributors. And at the end of the day, it also, like I said, comes down to the participant. And our participant is a wonderful person and a very hard worker, and he's curious and dedicated to the science and just very committed. You couldn't ask for a better person to work with.
And as Nick said, I think learning from scientific literature out there and previous studies has really helped us shape our direction, especially for a replacement. As Nick said, we have doubled the number of electrodes because we have seen with previous studies that the number of electrodes they had was not sufficient to give us accurate decoding of speech. So doubling the number of electrodes, targeting more localized areas in the brain that we believe contribute towards speech production really helped us to improve our system. And we also focused a lot in developing real-time systems. So our system works really, really fast. It works at 1 millisecond resolution, collecting the data, filtering it, processing it, and then which enables us to decode speech in real time and produce text or sound with very small latencies.
That is amazing. And let's talk a little bit more about that. About, first of all, the difference with the previous studies from Villette. It was the number of Utah arrays. Yes, I think in their studies it was 2, and in your study 4. Is that correct?
Yes, more or less. So the Stanford study with their participant T12, they did implant 4 microelectrode arrays, and they implanted 2 of them into premotor cortex, where they knew there would be these orofacial motor-related signals, and they implanted 2 more of their electrodes into the inferior frontal gyrus, which is it's a key hub in the language network, and on every speech-related fMRI you can take, it looks like you'd be able to decode speech from the signals there.
Also known as Broca's area.
Yes, yes. But when they built their speech decoder and they tried to decode speech, the neural signals that they've gotten from that IFG area weren't particularly informative for that purpose. So for their actual speech decoding study, they've only used the 2 arrays that are in their premotor cortex. And then now in the basic science that they're doing beyond that study, they're learning a lot from this other brain area. Just in the particular speech decoder design that our labs use, that area didn't seem to contribute very much.
And you made conclusions to double the number and implant them all in the premotor area. So what was your choice of implanting the electrodes? What areas did you target?
So Broca's area was supposed to be kind of language hub, which would contribute towards fundamental composition of language and things like that. But we have seen from Stanford study that it did not do so. But literature suggested that there is another interesting area in kind of middle precentral gyrus, which is known as 55P, which also contributes towards more language production and language preparation. So we thought it would be really interesting to target that area. We have never implanted a new tire in that area, so that would also be a unique opportunity to study what that area does. And premotor cortex really functions for speech production, and we have seen from other Utah studies how it contributes towards hand movement control. So this was a unique opportunity to target that new area of middle precentral gyrus, and we selected a replacement to go from the speech hotspot, which is ventral premotor cortex, and then go all the way towards middle premotor cortex. And we are covering a wide spatial area as well as doubling the number of electrodes. So that gives us opportunity to look at different brain areas, how they contribute towards speech decoding, production of speech, and also opens up further opportunities to study language perception, language formation, and other speech-related scientific discoveries.
And before the electrode implantation, there is always this procedure of mapping the brain, yes, finding that area. So I'm very curious about what methods did you use, especially for that middle precentral gyrus.
Yeah, so there's this project that is called the Human Connectome Project, HCP. And this, I don't know if they're like a group or a project, I don't really know how to refer to them, but the people involved in this study, essentially they did fMRI scans of, I think, hundreds of patients in hospitals or just volunteers. And then they sort of averaged all those brains together and mapped them to a common space. And through an HCP pipeline scan involves many different individual MRI scans, and some of them involve passively reading while your brain is scanned, or trying to say something, or trying to move your hands, or something. And through this very complicated pipeline, which I'll just summarize, you can essentially get a map of somebody's brain and with pretty good estimates of what areas are where on it. And so BrainGate has sort of recently adopted this HCP pipeline, making it standard for new participants that are enrolled into the clinical trial so that we can best target our arrays to the spots that we think are going to be best to record from. And I think that Stanford's participant, T12, was the first one to undergo this scan, and it worked well in T12. And we have now repeated that with T15 prior to the implant. So yeah, you kind of end up getting a map with X% confidence at each pixel of how confident you are that it's this area versus something else. And Then it's up to the expert neurosurgeon to map between that scan on a computer and the brain that he sees in the operating room when he opens the craniotomy. And there's a lot of cool surgical approaches to accomplish that, but I don't know enough about them to go on about them here. Anyways, yeah, this HCP pipeline really has been successful, at least in BrainGate so far, for targeting where these arrays should go.
That's a very interesting approach. So we already see several differences that you utilized when you learned about previous approaches. And you, of course, used 4 versus 2 arrays in the motor cortex and additionally covered or sampled from that middle percentile gyrus. You also compare your study, at least in your video, with the previous study by a group of Eddie Chung. And they use a different approach to sampling the signal. They use grids that they place on the surface. How do you think this affects the results of the studies? What do you think is the major difference between sampling with penetrating electrodes like you are using and grid electrodes?
We're prone to giving a biased answer, but I'm going to do my best to be neutral, unless you'd like to answer instead.
Yeah, I think it comes down to the resolution of information that we are able to record using these 2 different technologies. With our approach of using Utah arrays, we can really target single neurons, and there is no other fundamental kind of source of information rather than looking at individual neurons that and really highly targeted areas of the brain too. So we are targeting very localized areas that we believe to be contributing to speech control, the control of the speech articulators, as well as we are looking at very high resolution of information. So those 2 factors give us very high signal to noise ratio in the data that we collect that enable us to decode more intricate speech-related neural correlates. And I think when you look at the grids that are used in other studies, you look at more spatially diverse signal with more aggregate activity of maybe hundreds and thousands of neurons that lie underneath those sensors. So that kind of gives you a different set of information, maybe not that targeted to speech. So I think that is one of the differences that may have helped us to decode language better from our recording system.
Yeah. And that being said, I mean, there are a few advantages to the ECOG approach that the Chang lab uses. And I mean, one is that you don't have to do all this really meticulous targeting that we were just talking about with HCP, because as long as you know roughly where you want to record, you can put the recording grid on top of that. And the other is that because there's advantages and disadvantages to it not going into the brain to record. A disadvantage is that, as Maitreyee mentioned, you're not directly recording the activity of neurons, but you're kind of recording an aggregate of many with low resolution and from kind of far away. But an advantage is that this is just a sheet that sits on top of the brain, so it's very stable. It doesn't injure the brain, and it doesn't really move that much over time. So they, have demonstrated in their studies that for 100 days or something, you can get good decoding without recalibrating your decoder. And for a long time, I think that has been considered one of the key advantages of ECOG over Utah arrays. But we are beginning to show in our work and with our collaborators that with some clever machine learning recalibration tricks, you can overcome any of the instability that would have arisen from Utah array implants, the neural signals that you would record on them. So really, I think the short answer is that there's a lot to learn about both of these approaches, and probably the shortcomings of each might be able to be overcome with more engineering. So for ECOG, they're constantly making these grids denser and denser and recording from more and more sites. And it's not really known what the limit of that is going to be. So far, every time they've done it, signal's gotten better. So If they do it again, will it continue? We'll see.
Yeah, we've seen that in the recent study. They doubled the number of electrodes, and their decoding accuracy was almost equivalent to those of Utah rays, two Utah rays placed in similar areas. So, yeah, I think it's really dependent on the technology that we use and how that evolves over time, and which is the best technology to give best outcome at a given time point. So, yeah, lots of choices. To make.
Yes, absolutely. And how many electrodes does each Utah array have?
There's a few different models of them, but the ones that we use are 8x8 grids. So they have 64 electrodes, and each of those has a recording site on the tip of it. So when this is implanted into the brain, each electrode goes about 1.5 millimeters into cortex, and then you record from the one contact at the end of each channel. There's other arrays that are bigger or smaller, though, or have multiple recording points along the electrode. And then, obviously there's all of these new recording technologies being developed, like Paradromics is making basically an upgraded Utah array with many more channels and higher density, whereas Neuralink is doing this other approach where they sort of thread individual electrodes into the brain. So it's not so much of a, it's exciting to see what's going to turn out to be the best, the standard.
And it will also depend on applications. So different applications might require different types of recording technologies or neural interfaces, which would be more suitable to those applications.
Yes, thank you very much. Many of our listeners are curious about the artifacts that are associated with the Utah arrays because people are comparing intracranial recordings with the recordings from the surface of the head, non-invasive recordings. So what is the difference between the artifacts that we can see in your recordings versus non-invasive recordings? Maitreyee, maybe because you worked with both technologies.
So with EEG, the major challenge is muscular artifacts. So if you blink, you see that in EEG. If you move slightly, you see that immediately in EEG. Your heartbeat is also present in EEG. And the algorithms that you use to remove those artifacts might not be that stable and are quite complicated to apply them in real time for the kind of VCI use that we have in mind. But with Utah arrays, you don't see those muscular artifacts. So really different kinds of artifacts that we see from non-invasive recordings. With Utah arrays, we could see interference from power line noise. Sometimes we have this kind of non-stationarity in the signal. This could be because of the movement of Utah. So when you breathe, your brain also moves. And if the Utah rays are not implanted very snugly with the brain, going directly perpendicular to the cortex, you might see some micro movement in the Utah rays that might lead to some non-stationarities. So I think major source of artifact is non-stationarity over time. Your neural recordings change over time. Your neural tuning also changes over time. So for example, if you have a population of neurons that is really, really responsive to a particular task, after a month, you might see a different population of neurons responding to the same task. So yeah, I think one of the main challenges is recalibrating our decoders to match that neural shifts that might happen over longer periods.
Mm-hmm. And how often would you need to recalibrate? Is it with each session, each day, or there are more extended periods of time that you wouldn't need to do that recalibration? How does it usually work?
Yeah, the answer to that can vary participant to participant, or even day to day within the same participant. Our solution has been to just always be recalibrating in the background. So for brain to text decoding, the model is constantly being updated with recent data to attune it to whatever the most recent properties of the tuning in the neural data are. And this approach has been, at least for brain to text, very successful. And again, I mentioned there's kind of some differences participants to participants. There's a million factors that can influence this. So sometimes you see lots and lots and lots of this non-stationarity, minute to minute within somebody. A decoder might work or not. With our participant, things are pretty stable most of the time. And we think that may have something to do with the number of channels that we're recording from. So if there's a level of redundancy embedded in the signals across the 256 electrodes, it doesn't matter so much when one shifts, as long as the others are still picking up something that's encoded similarly. So that's one theory for it. But the real answer is it's not really understood completely why this non-stationarity is there or what is causing it. But what we do know is that we can implement software solutions to solve it, like I mentioned, with the continuous fine-tuning that we do.
Yes. And of course, the most common question is how long can these Utah arrays stay implanted in the brain?
So with our participant, we are more than 8 months after the implant right now. And the way it works in BrainGate is when a participant is implanted, they agree to be a part of the study for at least a year. And beyond a year, they can choose to continue, or they could choose to stop collecting data, or if they really wanted, they could choose to have all the hardware explanted. No one's ever chosen to have the hardware explanted. And I think the vast majority of participants really get a lot of value out of doing these studies and contributing to science. So they typically choose to stay enrolled within the study. So our participant has been implanted for more than 8 months, and there's a participant at Stanford and one at Brown who have each been implanted for more than 5 years, I think. Yeah. So the upper limit is not really known. And the reason for that is a little bit morbid in that people that enroll in these clinical trials are not healthy people. So, when you're diagnosed with ALS, for example, you have maybe 2 to 4 years to live on average. So seeing timescales much outside of that range might be a little bit atypical. Or similarly, participants that have paralysis or had strokes or something, there's a lot of health complications. And I guess what I'm getting at is that we haven't seen a participant keep an implant for 10 years yet. And one more side point to that is that these Utah arrays are sort of a old technology. They've been improved by the FDA since the 80s for implants, and they've been iterated upon a bit since then, but it's the same base technology. And I think at the end of the day, the field knows that the Utah array in its current form is not going to be the long-term solution of something that you're implanting into everybody. And that's the gap that companies that Neuralink and Paradromics and Synchron are trying to fill. Neuralink has poured a ton of money into engineering something wireless and with whatever other advantages or disadvantages that it has for the goal of reaching a market where anybody can get this implanted if they want to. And they and these other companies do all sorts of testing to show that theoretically these implants could last for decades. And it just remains to be seen if that's true or not.
Yes, thank you so much. So we discussed already what your study did differently in terms of methodology, the hardware, yes, implanting the electrodes when compared with other studies. Now let's get into the signal processing, which you also already mentioned that you did in a way differently. You really focused on real-time applications, like Maitreyee said. Maybe you can elaborate a little bit more on that. So what improvements, what innovations did you bring into your project?
So I think we'll speak about both the technologies that we have, brain to text and brain to voice. Yeah, so this is really very new approach, and we have really not seen previous demonstrations of this brain to voice approach for speech neuroprosthesis. So we had lots of challenges to overcome. Our priority was always to get neural data processed very quickly with very little latency. So as I mentioned before, we did all the signal processing and noise removal very quickly within 1 milliseconds of acquisition of neural data. So that allowed us to spend a little bit more compute time on the decoder, which would actually decode that data. So there are different ways when you decode speech from neural data. The first one is data acquisition. We record the data from the hardware. It goes into a computer where we denoise the data, we filter it, and we extract useful features from that data. So we filter it to remove ambient noise, low frequency noise, other things, other artifacts, and then extract features. So with single neural resolution of data, we can get action potentials, and the proxy to that is spikes. So we see how many times a neuron has fired in a particular time bin. So those are spikes that we get. And then we also look at the broadband activity, which gives us more broader view of the neural data that we have collected. So these are the features that we chose for our decoders. And all of this processing was done within 1 milliseconds in our pipeline. Then with these features, we send them to the pre-trained decoders for brain to voice and brain to text, and then they synthesize either voice or decode phoneme sequences again in real time. So for brain to voice, our challenge was to get the output with minimum latency such that it sounds like your own voice. Because when we speak, we hear our own voice with very little latency. And if you have that increased delay, it won't sound natural to you. You'd it wouldn't close the loop. So we were focused on closing the loop and enabling this real time speaking and conversation with auditory feedback. So the brain to voice pipeline worked at a resolution of 10 milliseconds. So every 10 milliseconds, you would produce a 10 millisecond window of voice. So it was instantaneous production of voice. And then we'd send that to the speaker, which would play it back to you. So that also incurred a little bit more delay. So to give you an example, it sounds like when you speak on a microphone and you have a speaker at the back of the room, you have this little bit delay, but when you speak, you still hear it back. That is the kind of latency that we have in this system. And with brain to voice, one of the major challenges we have is not having the grammatic speech again to train the decoder. So our participant speaks, but we don't know what he's saying, how he's saying each word, how he's spacing his syllables, etc. So we also worked on a novel algorithm to kind of create this target speech, which would help to train this decoder and produce speech in real time. We are also investigating other aspects of speech, so how we can decode pitch from the brain activity or intonations or volumes, which could make this even more natural and more expressive moving forward.
Very interesting. And what helped you to achieve these results, to minimize this delay time to basically 10 milliseconds? Yes, and have this very high resolution.
It was designing the whole signal acquisition processing system. So we have a big card with 4 different computers doing 4 different tasks very quickly. They have high compute processors on them. So that definitely helped. We also used intelligent algorithms to minimize the compute time. So reduce the time complexity of the algorithms, as you call it in computer science, and really cleverly design the system so that these decoders work super fast. And that has also posed some limits on what the decoder architecture could be. So we could get better decoding, but we wanted to get it fast enough as well. So we had those considerations in mind to get those outputs as quickly as we can and try to push this boundary with accuracy as well as speed.
I just want to add that Maitreyee was so good at optimizing the signal processing pipeline and her decoder and making them so fast. She was so good at that the biggest hurdle in latency has just been playing the sound out of the speaker fast enough after it's generated, which is a really silly problem.
Yeah, that's amazing. And Maitreyee, you also mentioned denoising the signal, the first pre-processing stage, and you mentioned removing low frequencies. So what frequencies did you concentrate on for further analysis?
So what we are looking at is called spike band spike band is higher frequencies above 250 hertz up to 5000 hertz. We can look at spiking activity in that band. So we're looking at spiking activity as well as broadband activities. So spiking activity is really localized to individual electrode that we are recording from, but broadband activity gives us more robustness and more stability because it kind of looks at somewhat more aggregate activity around that particular electrode. So using the combination of these two features, we are able to enhance our decoders.
So when we're decoding, we're only looking at signals above 250 hertz. But obviously, there's lots of fields that get a lot of value out of neural signals slower than that. So we also save those other signals. We save the whole broadband thing, and then we, can look at that offline, and maybe it'll make decoding better. Who knows? But for now, above 250 is what we use.
Thank you. And Maitreyee, you also talked about looking at other aspects of speech, such as intonation or volume. Did you notice any features? Were you able to extract any features related to those components?
Yeah, we are really in early stages of looking at those paralinguistic features, but we believe that if we are able to decode them reliably enough, they would definitely contribute towards better speech synthesis and really improve the overall experience of using this BCI for naturalistic speech.
That is amazing. Yeah. Nick, do you have anything to add for your part of brain to text in terms of algorithms and signal analysis?
Yeah, I can. So really, I use the same neural features that Maitreyee described for the brain to text decoding pipeline. But I can add that, of course, brain to voice latency is a huge deal. And playing that audio back in real time makes a big difference. For brain to text, you have more flexibility with the latency, which gives you a lot more flexibility over the model architecture and how frequently you're going to be making predictions and that type of thing. We still want to keep the latency relatively low so that words are appearing on the screen as he's speaking. But that can be hundreds of milliseconds potentially rather than 10 milliseconds like she's describing. So there are, different considerations to make depending on what you're trying to decode and how it's going to affect the participant in this closed loop scheme.
And I believe in brain to text pipeline, language models also help in filling the gap or the errors that happen in the decoding to really improve the performance and make the system usable for the participant.
Yeah. So as Maitreyee alluded to earlier, she is causally predicting voice from brain signals, whereas with brain to text, we have this huge advantage of using language models because the English language is, it has statistical structure. When somebody says 3 words, you can make a pretty good guess what the next word's going to be, or you can fill in gaps or all types of things like that. So, although we are predicting text in real time and updating it on the screen, we're also non-causally looking back at all the relationships between the words that have been predicted so far and updating with our best guess. And then at the end of a sentence, when we know we're done and the participant presses the done button to finalize, we have more time to work with there, and we can really leverage these large language models, which is another exploding field. We're fortunate that it's exploding alongside speech neuroprostheses because for brain to text, they're very useful. So what I'm saying is at the end of a sentence, you can sort of take this more expensive, it's called a rescoring step to reanalyze what all of the possible things that the participant may have said there based on what you decoded, and then coming up with the most valuable answer. And that step can also be personalized a bit toward a participant. So, or toward a conversation where there's existing context. So, what you said 3 sentences ago might be influencing what you're trying to say now, and we can leverage that information. And then also, some facts about the participant, like their day job or their family members' names or where they live, things like that, that can also heavily influence what they might be talking about. So incorporating all of this contextual information is another big advantage of this text decoding scheme. So this is why Maitreyee has rightly said that brain to voice is much harder. You don't have any of those contextual cues to incorporate into your decoding pipelines. You just have to be super good at decoding neural signals.
Yeah, there's no corrective mechanism because once you play the sound, it's out there. You cannot go back and correct it based on what was said before or after and make it more intelligible. So that is one of the major challenges that we are working on. I also wanted to mention that previous studies have used kind of discretization of voice. So, for example, Chang 2023 study, they also tried to synthesize voice from their brain signals, but their approach is quite different to ours. So they're discretizing all the sounds into some fundamental units of sound, but they're again discrete units that they're stringing together the sequence to make the sound, whereas our approach is to output sound in continuous space. So the participant can literally say anything any way he likes and would like to synthesize that voice without discretizing it to sound like a particular word or something else. So this also poses additional challenges because your output space is infinite. There are infinite sounds that you can make and infinite ways in which you can say a word. So every time you say a word, you'd say it differently. It's not going to be the same. So those are the kind of challenges that we're dealing with to produce more naturalistic sound.
Thank you so much. And what was your participant mostly interested in communicating? What did he want personally to communicate through the interface you provided?
I think I can give you a little bit more context about his day-to-day life to inform. So for ALS, he was a very talkative, funny person. He said that he could just speak for hours and hours and, it's just, not think about it even at all. He just had a lot to say. Since his dysarthria has sort of set in with ALS, obviously that's been very much taken away from him. He can communicate with his loved ones, but only through a lot of effort and frustration and patience. So most of the time, day to day, what he communicates has been limited to like essential transactional things like, can you do this for me? Or I'm uncomfortable, or please do, so he's not really speaking for the sake of communicating, like beyond basic needs. So with a speech neuroprosthesis, and when he decided to enroll in this study, he's also very competitive. He just, well, first of all, he wanted to break all the records, and second of all, he wanted to get his voice back and his sense of humor back, and tell jokes, talk to his daughter, talk to his family members, talk to people at work. I mean, he still works full time. He wants to be able to more easily communicate with people in that environment. He wants to like, I don't know, make fun of people on video games, things like that. Like just any little thing that we take for granted with communication. I think you could say he wants to be able to do that. So
And it has been incredibly useful to us as well because he communicates with us using this system because we cannot understand his speech without an interpreter. So when we go to the sessions, we just turn on the system, he speaks to us, we talk to him, and that has really also helped us research and get feedback from him on how different tasks have worked for him. So yeah, it's really an incredible system that he can really use to communicate with people.
So, what was his feedback on the process and on the results that you were able to achieve?
well, we asked him if he knew it was going to work this well, or if he thought it was going to work this well. And he said, of course, yes, of course it would. He's him. He always wins, that type of thing. So, I mean, of course, he's grateful and he really marvels at this technology. He does not take it for granted, I guess, is what I'm trying to say. I think he was on some level surprised at how well it worked, whether he wanted to admit it or not, as were we all surprised. And seeing immediate utility out of something like this was amazing. So something I didn't mention was in this second research session where he used this very large vocabulary to, speak these prompted sentences. At the end of that day, we also gave him the opportunity to just try to say whatever he wanted and see if we could decode that instead of him reading something off of a screen. So we only had a little bit of time left at the end of this day, but we were able to do 10 sentences, and he used all 10 of them to talk to his daughter for probably the first time really in years, because he hasn't been able to speak in that's the only way he can communicate with his daughter. So asked her how her day was and said he was happy to talk to her and he'd been waiting for this for a long time and those types of things. And so I think that was really an immediate demonstration of how useful something like this could be in anybody's life who has this type of disease, but also specifically in his.
Yes, absolutely. And what happened after that second session? Because you are referring mostly to first and second session. What was going on after that? What type of studies were you doing? What type of communication did you explore? And how did it work?
Yeah, so we have several ongoing projects. And I mean, obviously, with both of these decoding projects, we continued to collect a lot more data. But for the BCI award, we wanted to just limit ourselves to this initial impact and, just show like, wow, this is great. This can work immediately. That's huge for the field, right? So for the Brain Detects project, we are working on publishing it right now. But I can say that, we've continued to collect a lot of this data. We've continued to decode speech, and he has actually began to use this system just for his day job, 2 days a week right now. So for 2 days a week, we initialize the system for him, and he can use it for 8 hours or however. I think the current longest continuous collection right now is 8 hours of decoding. And he just uses it all day. He does Zoom calls and Slack calls. He sends emails. He talks to visitors. He cracks jokes, that type of thing. So that's been really exciting for us and for him, of course. And we're working on publishing those results right now.
We're also looking at different fundamental science questions about how speech is encoded in this part of the brain, and then what other information we can get from it. So we have been very busy for the last 8 months, and we're investigating different kinds of questions. And yeah, hopefully we will get to see much more that comes out of this study.
Yeah. And my question also is how tiring is this process for him? You mentioned that he can work, for 8 hours. So from this, I would guess that, he feels pretty good. But did it take time for him to get used to the system? So how did it all develop until yeah.
I think over time, he's found a strategy that is minimally exhausting for him, but still results in very good decoding quality. So, for example, if we asked him to speak or to attempt to speak overtly and with volume continuously, that can become very tiring for him. But if he uses a much lower volume, it's kind of a volume that you can't even hear that he's speaking, but he says he can hear himself speaking. Using that approach, he can go for 8 hours or more. The limit in that 8-hour case was not him. It was just the end of the day. So I think for every participant, though, the answer to that is going to be very different based on their condition. And in his case, this is just what it is right now.
And I think for our participant, this is less effortful than communicating using his own speech, because it is really hard to understand him, even by expert listeners, as Nick said. So if he has to say something, he has to repeat the same thing 5 times, 10 times, which can be exhausting for him. So this gives him an avenue to communicate very quickly and very fast.
Mm-hmm. And I'm also curious, how much time does it take from the beginning, when he just gets to the lab, until he can already communicate through the system?
Well, so first I want to note that we do all of our research collection at his house. So we have a setup there, and that's a little bit dependent on the day, but normally the answer is very quickly. So we may find that we plug him in and instantly decoding is as good as it's ever been. And other days, like what we talked about with this unpredictable non-stationarity, other days it might just take a little bit longer for calibration to happen, but it's not that much longer. It's like within 10 or 20 sentences, things will begin to settle down, and then he can use it for the rest of the day. But from when we get there, we just kind of have to like turn on our system and set it up. We have to hook up the recording hardware to his head and plug him in. And there's a few like short initialization steps that we have to do just to get things running. But all in all, I'd say that takes under 15, 20 minutes, something like that.
Yeah, thank you. Can you summarize the results of this initial study?
I'll summarize it for brain to text, and you can summarize it for brain to voice. So, in a 45-year-old man with ALS, we implanted 4 microelectrode arrays into his precentral gyrus. And on the very first day of recording his brain signals, we were able to decode what he was trying to say out of a 50-word vocabulary almost perfectly. And on the second day, when we expanded that to a very large vocabulary, we were able to decode what he was trying to say with more than 90% accuracy. And he was even able to use it to speak to his daughter and say unprompted sentences that he had wanted to say to her for years. And in the time since then, we've collected a lot more data. We've optimized our algorithms and made enhancements. Things have gotten better accuracy wise, and he now uses it multiple days a week to do his job and talk to his family and his loved ones.
Okay, so with the same electrodes and with the same participant, we have also been able to synthesize his voice in real time with real time feedback, which is a step closer towards restoring naturalistic speech in a person who have lost their ability to speak. We have had to overcome challenges because the participant cannot speak intelligibly, so there's no ground truth speech. And then with voice synthesis, the latency is also a major issue. So overcoming those challenges, we have been able to synthesize his voice, which is nearly intelligible.
Thank you so much. And what would you consider the limitations of your study upon which you want to improve in the future?
So in these types of studies, we're always limited by the number of participants, obviously. So this is an N equals 1 study, and we're really interested to see if this high performance speech decoding can translate to other participants, either other participants with ALS or perhaps other conditions.
Yeah, in terms of voice synthesis, we are still not fully intelligible. So there is a lot of progress to make. We have shown the first step, but it needs to improve. Whereas brain to text is at advanced stage where it can be usable in his personal life, which is great. But again, as Nick said, we'll have to replicate the study and these approaches on multiple participants to kind of make it available to wider population.
And I'll point out that although the brain to text performance is very good, there's still always room for improvement. So I can say, as we talked about language models earlier, which were, harnessing the statistical structure of the English language to make predictions. A failure point of that is proper nouns in some cases. So there's many sentences where you might put a proper noun, it could, first of all, it could be any other proper noun, or it could just be a normal, like a noun, for example. And if you ask a language model, what's the most likely word that should go there? It's not going to be a specific name. It's going to be some other word sometimes. So there are some small things like that we can always improve for this type of decoding. And then, of course, when it comes to clinical translation, people don't really, they're not going to want these wires poking out of their heads, and they're not going to want a closet-sized computer cart sitting next to them the decoding. So there's a lot of work for the industry to do to perfect these types of recording systems and make them into little embedded computers. Maybe it can run on your phone someday.
Yeah, and even though we have been conducting this study for the last 8 months, we still have to see the long-term stability and long-term accuracy of this decoder. And once you undergo neurosurgery and implant these electrodes, the expectation is that the system will work with equal accuracy for several years to come. And that is also going to be another challenge. And then the disease progresses, it progresses differently in different people. So we have to see how that disease progression affects the brain computer interface that we have built. And eventually, people with ALS or degenerative diseases lose their ability to speak altogether or go into locked in states. So that might also pose other challenges. So there are many open questions that we want to investigate. And yeah, hopefully doing these kind of trials and more participants over longer durations will help us answer some of those questions.
Thank you. And what was the biggest challenge for you in this project, and how did you solve it?
One of the biggest challenges, I don't know if it was the biggest challenge, sort of a blessing and a curse. When you join a brand new lab, you have to build everything from the ground up. So the good part of that is you can make it the way that you want it. And the hard part is that you actually have to do it. So we were, fortunate enough to have, I think we both joined the lab and we had about a year before we recruited a clinical trial participant. And in that year, we spent a lot of time on perfecting the system. And it's still not perfect, but I'd say that's one of the biggest challenges that we've faced. And the solution is just time and effort.
Yeah, and also working on the projects that do not have any prior or any starting point. So brain-to-voice, again, this was never done in a person with ALS who cannot speak before. So it posed its own challenges to come up with a completely new approach and solve all those challenges and come up with techniques that would work in people who are losing their ability to speak. So I think, yeah, many algorithmic challenges, scientific challenges, engineering challenges. Yeah.
Yeah, yeah. And Maitreyee, you said it was the first time this type of approach was used. How did you come up with this idea? Why did you decide to create this brain to voice interface?
I decided to do it because it has not been done before, and it was a unique opportunity to explore something very new. It was a great platform because we had already decided that we would have a participant with 4 arrays in speech motor cortex. So that is the highest resolution of information that anybody has ever gotten from speech motor cortex. So this was a good opportunity to test this new system and kind of think about solving the unique challenges for brain to voice synthesis and come up with different algorithms, test different systems on the data that we had from our previous participants, see what works, simulate some data, and try out different decoding approaches. So, yeah, it was a lot of thinking, a lot of contemplation, and a lot of building.
Just to explain it better to our listeners, that the previously conducted studies, they can convert brain signals into text and then from the text into voice. Yes, just synthesize speech. But in your project, it's for the first time the voice was synthesized directly from the brain signal. Is that correct?
That is right. And also to clarify a little bit, there have been some studies where the voice was synthesized from brain signals, but it was recorded from participants who could speak. So it was mostly ECOG studies where people undergo epilepsy monitoring. So they were in the hospital, which gave researchers opportunity to record their data, and they were overtly speaking intelligibly. So you had ground truth for the neural data that you recorded. Also, this was never done in real time before. So you couldn't hear their voice being synthesized in real time as they're speaking. So two major innovations here are synthesizing voice in a person with ALS who is severely dysarthric, so we cannot understand what he's saying intelligibly, and also producing this voice in real time and playing it back such that it kind of recreates this whole human speech motor loop.
And what was the most unexpected part of this project for you? Maybe some results, maybe some approaches, anything that you didn't expect before you started the study?
I think I have a good one. In our first research session, I've already mentioned, how many things can go wrong in this type of thing. But I think one of the most surprising parts of that day were after we had collected some of just the baseline open loop data to train our models with, we were taking a little lunch break and I was training my brain to text model on one computer and Maitreyee was doing her brain to voice one on another one. And I remember just seeing for the brain to text, just seeing that there were perfect predictions coming out of the model on like held out data. And I looked over to Maitreyee and she was like, hers had never looked that good either. And she asked me if something was wrong, like if somehow our data was contaminated or something. That was a good
Yeah,
moment.
it was an exciting day. We were able to demonstrate these two decoders on the very first day, and everything went according to the plan. So, yeah, it was a great start to the clinical trial, which will continue to run for many months and years.
Yeah, and it's not so often we have everything running according to the plan. I think it's more exception rather than a rule.
Broadly, it worked according to plan, but as with anything, little things go wrong and it's just about fixing it quickly.
We had many, many challenges occurring over the last 8 months. And here we have gotten used to being very quick on our feet to debug what is happening, troubleshoot on the spot. And we have an entire team to support us. So we have grad students, uh, research assistants, and of course our PIs who are there to help us. We are constantly in contact with them when we run our sessions, and then people help us troubleshoot and kind of make this happen. So this really is a team effort.
Absolutely. Yeah, that was one of my questions that I haven't asked yet about your team and its contribution. So thank you for mentioning all of that. And what do you think became possible with this study that wasn't possible before?
For the brain to text, I think that answer really writes itself. I mean, the participant is using it to speak. It's his preferred way to communicate. And I don't know how much better you can hope for than that, right?
Maitreyee, do you have anything to add?
I think great participant who is invested in our research and is willing to try out different tasks, which gives us opportunity to test different approaches day in and day out. That has been really useful, and also getting this level of neural data, which was never done before. So I think this opens up many more opportunities to answer more difficult questions, which we could not even think of because of the lack of high resolution neural data. So all of these things, yeah, really are very unique to this study.
So, what are your next steps now? What are you working on?
Yeah, we want him to be able to use the system completely independently as much as he wants, whether that's 1 day a week or 7 days a week or whatever. So there's a lot that goes into that from an engineering perspective of making a system that's robust and usable easily. So that's one thing that we're working on. Another thing that I'm working on is we have this speech decoder that we know works well, and it's based on machine learning. So it's kind of like this black box, right? You know that speech is embedded in these neural signals because you can decode it, but you don't actually know exactly what the decoder's doing. What types of speech features are actually being encoded in that neural data? So that sentence describes 1,000 projects, but one project that I'm working on is trying to take a basic science approach to understand what are these, how are phonemes encoded in the neural data, and how does the decoder take advantage of that? And understanding that can help us build better decoders that leverage this tuning in a more informed way.
Thank you. Maitreyee, anything you would like to add?
I'm working on improving this brain to voice decoder, and we want it to improve to the level that he will be able to use this system for his personal use as well. And also, as I mentioned, speech is really multidimensional and it has lots of other expressive biolinguistic aspects to it. So I working on whether we can identify those from the data that we have would be a next challenge, and embedding those to produce naturalistic speech. So that is my aim.
Thank you. And how do you see the field of speech neuroprosthesis, let's say 20 or 50 years from now?
I hope by then there's a reliable, like a product, a solution for anybody who needs something like this to be able to communicate just by attempting to speak. And so beyond that, I imagine the field is further perfecting those technologies and also just trying to expand it to new patient populations, like people who are completely locked in, or people with aphasia, or people who never spoken before, maybe extending to cognitive aspects of speech. Speech is this thing that's kind of motoric, but kind of cognitive as well, right? So I think there's a lot of directions to go in, but I hope that in 20 or certainly in 50 years, the people who need a device like this to help them communicate have it available.
I'm also more and more confident that we will have such a device in the near future. But one of my major interests is also to see non-invasive technologies being developed, low-cost technologies being developed to offer an alternative solution to these high accuracy, high resolution systems, because having invasive system in any form is going to be more expensive and may not be available to everybody around the world. So the focus of my research in the future will also be to develop some of these technologies, maybe simplified features using non-invasive brain-computer interfaces. And we have seen this start to speech decoding from non-invasive signals, and people are getting interested in it. So I'll be really curious to see where the field goes in non-invasive BCIs as well for speech decoding. And hopefully we will also have an equivalent, maybe simplified product for people who want to use non-invasive technologies, wearable technologies, and more affordable technologies that can be made available throughout the world.
Yes, I think that's very important to develop those technologies. And what do you think those technologies will be? Will it be EEG based, NIRS combined with the EEG based? Now there are also new portable MEG devices that are being developed. So what do you think will be that technology that would allow for speech neuroprosthesis that can be used non-invasively?
I think the bottleneck for that would be the hardware device, wearability of the device, and the resolution of information you get from those. So maybe combination of these might be useful. Yeah, we are really limited by signal to noise ratio from non-invasive technologies that we have currently, but I'm sure that with more engineering effort towards developing better sensors, we could overcome some of those limitations and start getting better quality data so that we can do much more with it.
Thank you. And what do you think are the three main skills for each of you without which this project wouldn't be possible?
Yeah, this neural engineering seems like you have to know a bit of everything, right? Since coming to this lab, I've picked up a lot of skills that I did not have before. But if I have to name three, neural signal analysis is a key one, understanding not only the biology behind it, but the techniques to analyze that data. The sort of engineering side of this, like developing something for real-time use and getting it running on a computer in real time, and maybe even building the computer or putting it together. It's sort of maybe the computer engineering side of that is currently a huge part of this. And I think at the end of the day, in any scientific field, you have to be good at presenting your research, whether that's writing it or communicating it. You can do the best work ever, and if you can't tell somebody about it, then it's not very valuable.
Yeah, no, you did it today perfectly. You communicated absolutely amazingly. So you do a very good job with that. Maitreyee, what is for you?
Everything that Nick said, but I'll also highlight some of the higher level skills. Being able to learn different things very quickly, because you have to read new scientific literature, understand it, and then keep an eye out on new developments and machine learning. So there are like a lot of things going on. So you have to be able to learn those quickly and be flexible to change your approach, question yourself constantly. Many times it happens that you come up with an idea and you stick with that, and then it becomes really hard to kind of look slightly from outside and see if there are other approaches that might make this better. So that, as well as get transferable skills, which are really useful. So you learn from one domain and try to apply them in another one. I think my multidisciplinary background has really helped me to tackle this project. And yeah, we're learning new things every day, which is really exciting.
Yeah, I think for someone trying to get into this field, whether they're already in something related or not, the multidisciplinary background is super useful, because if the only thing you know is neuroscience and motor neuroscience, you might not be able to think of a new idea that already exists in another field in some way and translate it. And I think a lot of the people in our lab and in BrainGate as a whole and in related labs, they're very multidisciplinary teams, and that's sort of where the strength comes from.
Yes, absolutely. And today you already mentioned synchron, and this is example of how the idea from different field was integrated into neurotechnologies. Yes, so the many actually of our guests, of our podcast guests, are emphasizing that. So thank you for mentioning it. Very good. And then what would be your recommendations for those who want to get into the field of neurotechnologies and do this type of amazing work that you are doing?
I think in terms of studying, having fundamental understanding of mathematics and machine learning and neuroscience is really key. And also exploring research opportunities early on in research labs in the universities or industry that would also benefit in getting exposure to neuroengineering and latest developments in the field.
Yeah, and there's a lot of labs that don't exactly do BCI work that have probably 90% overlap of like the kind of things you need to know to do BCI work. So studying population neuroscience with primates, or studying non-invasive approaches with humans, or things like that, they're all equally important. Something that, especially recently in this field, I think has become super important is the ability to code. Now with machine learning, especially, it's a very important skill. I think that if you had to choose one thing for making a brain computer interface, you have to know how to code, right? Or you have to know how to learn how to code or something, right?
Yeah, ability to learn new things.
Yes.
Yeah. And also, usually we say that it's important to learn how to assess big data. But with neural engineering, especially with clinical trials, we have very limited data. So,
Small data.
yeah, small data. Yeah. So, working on approaches to build different technologies that use limited amount of data will also be really useful.
Mhm. Thank you. And you applied for BCI award, obviously, because you won. What prompted you to do that? What did you decide to submit your work for the award?
I think after our first week or two of data collection, we were like, well, that went pretty well. We should apply for this. It was all pretty short term because it was not long before they were due that we were collecting the data.
Yeah.
But I think in our case, it was a pretty simple decision.
And we're excited to share our work in any form. So this was a great platform to share it with the community who's really interested in BCI. And we have seen so many wonderful studies come out of finalists and BCI award in the past years. So yeah, we thought it would be great to kind of contribute to that and share our work on this platform.
Yeah, it was really exciting to not only to apply for it, but obviously to win it, because, as I said, we had spent the first year or so of our time in lab not getting any data, just preparing to get data. And so to get it and have it be good and worthy of a submission so quickly was very exciting.
Absolutely. And what would be your recommendation for those who are still on the fence and thinking to submit or not to submit?
I think they should submit because, the practice of presenting your research into a demonstrable package was a good exercise as well. So, and also you get an opportunity to share what you're doing, which is always exciting to researchers. So, definitely submit if you think it is exciting and people might be interested in it. Go for it.
Yeah, I think worst case scenario, you're going to make some new connections and meet other people in the field and probably learn a thing or two. So it's definitely worth it.
Absolutely. Thank you. Can you think of anything in your life that you thought is impossible? Maybe even in this study and research, it doesn't matter, that you did in any case, you still did it and you proved that it's possible. And how did you do it?
That's a hard one. Maybe instead of impossible, I'm going to switch it to difficult.
Okay, let's do it.
I'd say for me, I grew up in Pittsburgh. I went to all my school in Pittsburgh, including undergrad and grad school. For my whole life, I lived in a 2-mile radius. And suddenly I was moving 2,500 miles away to California to start a postdoc here, hoping it would go well, hoping I wouldn't mess up the opportunity somehow. And I can't have asked for it to go any better than it has. So I guess that's my answer.
Yeah, and I'm sure it was challenging for you to make this step. And and it went well. So it was worth the risk.
Yes.
Yes. Okay, very good. Maitreyee.
When I was a child, I did not think that I'd get opportunity to be a scientist and work in these different prestigious universities with such wonderful collaborators across India, UK, and US, and kind of really work on these multidisciplinary technologies. So that has been really great. It's a lot of hard work, but I'm also very grateful that I get to work with clinicians, patients, industrial partners. Some of my previous work has been commercialized. So I'm really getting to learn from all these people and different stakeholders in these studies. So that has been great. But as a researcher, you always also face a lot of failure and you have to keep trying, and success doesn't come that easily. You have to keep trying and work on different things and not lose the motivation to work on the problem at hand.
Yeah, I think that point about failing a lot is important. Sometimes people assume that the best scientists are just perfect and they get it right the first time. And no, what you see is the 1,000th iteration usually, I think. And it's often hard work and commitment and a curious mind that gets you there.
Yeah, lots of failures and lots of rejections as well. So winning this award is great, but yes, a lot of rejections throughout my research career. So yeah, it's exciting to work with such driven people and such an incredible group. We constantly learn from each other. We collaborate very closely, and I think that really benefits our team to build these kind of new technologies and really push the envelope of research.
And I think it should encourage more big collaborative research groups like this. And I think to some extent it has. So like Pitt and Chicago have formed their, what are they, cortical bionics now. And I think in academia and in BCI specifically, perhaps, that this consortium model is really beneficial for everybody, from PIs to participants to neurosurgeons to trainees. And I think traditionally academia is not really like that. It's like every lab for themselves. So there should be more collaborations.
Yes, absolutely. I agree. And Maitreyee, you said that it's very important, even through the failures, to still keep being motivated and persevere. So, what keeps you motivated? What helps you, even in spite of all the challenges and failures, still move forward?
It is, you have a goal and you want to achieve it, and you are prepared to work towards it. Everybody knows that success doesn't come easily. There is hard work, but even after hard work, you might not see the success. So I think just being persistent, trying to learn new things, trying different approaches, looking out of the box to solve the problem that is really difficult. I think all of those things kind of motivate you on day-to-day basis, but it is also a lifelong struggle. So yeah, it's both. It's kind of helps you motivate because you're working in a really cutting edge field, which is making incredible progress, very rapid progress. So you see all the great developments that are happening around you. That motivates you. Sometimes you get a great idea in a shower and then you're excited about that for the whole day. That keeps you going. And then again, you have these opportunities to present your work. You get feedback. So, for example, we presented at conferences and this work was really well received. So that motivates you. So these are all different little things that keep you going. And then sometimes you have a great brainstorming session with your team and your PIs. So that helps you be motivated to kind of continue work.
Thank you. And Nick, is there anything for you that keeps you motivated throughout failures?
I like the way that our participant puts it of just being stubborn, just not taking the failure as an answer, just trying more. But no, I think a lot of things that Maitreyee said resonate with me as well. And your environment makes a big difference. So having a good group of people around you to help motivate toward this goal and kind of if everybody's working hard, it's hard not to work hard, I think is a way to put it.
Very true.
Yeah. Thank you so much. Thank you for so many insights, for all these wonderful things that you described and all the great suggestions you also provided our listeners. As we're nearing the end of our podcast, is there anything else you want to share or to tell our listeners and future readers, because this also will be published in a book?
I just want to say thanks for having us on and for having so many insightful questions prepared. It was fun to talk to you.
It was my great pleasure. Thank you, Nick. And Maitreyee.
For having us. Yeah, it was great talking to you. And yeah, I'm really excited about where the neuroengineering, PCI, neuroprosthesis field is going. And yeah, I look forward to all the wonderful discoveries that are made in the coming year.
Thank you. And of course, I wish you all possible success in your study, in your future careers. And I hope to see you maybe talking to our listeners several years down the road to see what have you achieved and what are your next steps. So to continue to see your journey and how you are doing. So all the best. Thank you.
Yes. Okay. Thank you.
Dear Neurocareers podcast listeners, thank you for joining us on this incredible journey into the entrepreneurial world of neuroscience and neurotechnologies. I hope you've been inspired by the stories of those who are turning groundbreaking ideas into impactful realities. If you are looking for more guidance on succeeding in neurocareers, book a free consultation with me, your podcast host, Dr. K, at the Institute of Neuroapproaches. So, what are you waiting for? Let's navigate the path to success in the world of neurocareers and make the impossible possible together. Are you a fearless pioneer in the world of brain-computer interfaces? Do you have an amazing project that's pushing the boundaries of what's possible? Then we want to hear from you. The International BCI Award is one of the biggest and most prestigious awards in the BCI world, and the deadline for submission is September 1st. But don't worry, you don't need a top-notch budget or equipment to be a nominee or a winner. All it takes is a great idea and the determination to make the impossible possible. So, submit your project now at bci-award.com or find a link in our podcast notes and join us on this incredible journey to explore the world of BCIs. And make sure to listen to our upcoming BCI Award Neurocareers podcast series for tips and tricks on creating a successful submission. Who knows? You might be the next nominee or winner of the International BCI Award and proudly showcase your nomination on your resume to impress potential employers and establish a successful career in neurotechnologies. But don't stop there. If you are looking for more guidance on succeeding in neurocareers, book a free consultation with me, your podcast host, Dr. K, at the Institute of Neuroapproaches. So, what are you waiting for? Let's make the impossible possible together.